Hi, everybody. Very glad that we are here today with you on this webinar. We will guide you through some of our newest results when it comes to iPSC culture, cultivation, and let me just quickly introduce ourselves before we jumping into the topic and some words losing about our company. I'm Patrick. I'm a biotechnologist here from Germany with a background in chemical process engineering. One of the inventors of the membrane stirrer, which we will lose some words more about later on, and the CEO and co-founder of the company BioThrust. With me and our process expert for iPSC cultivation is, as already mentioned, Marco. He has a splendid 10 years experience in iPSC expansion and differentiation. He is responsible for all our internal and external partnerships when it comes to iPSC development. And yeah, well, he's also not Guinness World, but world record holder of the highest published 3D iPSC density in our bioreactor. So before we showing the biological approaches, I just want to shortly dive into the company and our technology per se. So BioThrust was founded 2022. So quite a young company, as you want to say, with nearly 30 employees here in the most western city of Germany. And we are based on three technology pillars aside the splendid team we were able to gather here. And this is the membrane stirrer technology. It's the in-situ perfusion and controller systems, which we also show later on. We do with REPROCELL, but also directly sales across the whole globe, and already having partnerships on the North American continent and in the Middle East. So you guys may know better than I do, but the iPSC market is quite a difficult field to work in. It's steadily maturing. Yeah, we know that so more and more iPSC trials go into the field. Japan already approved the first therapy this year, which is a big step forward. Yeah, more than 100 trials are already in the pipeline. But also we had a lot of drawbacks when it comes to the restructurization of big pharma companies. Anyhow, we see a kind of linear growth in the market, nearly hitting $2 billion this year. But why is that not a hockey stick curvature? Why is this just linearly? And not just we think this is also depending on the scalability of those production systems, and that we are stick to somewhere in the middle between 2 and 10 liters. At least this is one of the reasons we figured out. So normally we have the classical upstreaming process, followed by the downstreaming fill and finish. By upstreaming, we have to deal with the source, and then what we will focusing on today is the expansion. We also did something in differentiation, and Marco will also comment on this later on very shortly. I'm very happy for questions from you. But this also with regard to the timing, we will look mostly on expansion today. So normally you have the classical trade-off between process yield and high cell quality. Yeah, you can stay in flask, or you can try to go to stir tanks, but either of one will impact highly the other. You can try to scale out, or you have to scale out if you're working in small volume systems, or you have to work with workarounds to somehow make it even possible to get to the cell numbers you need for your therapy. And this is cumbersome and didn't work so far in the past. So we now offering a system which is focusing completely on the yield with high quality, which is truly linear scalable from 300 milliliters to what we can already say 2 liters and beyond, and which is one integrated system which does not need external gas exchanging systems or nutrient supply. So when we have a look on the commercial efficient iPSC culture, we normally need low shear force or closely controlled shear force environment. We need a sufficient gas transfer, and most systems lacking here. And it's not just oxygen in, but it's also carbon dioxide out, which becomes a problem in bigger scales. We need homogeneous mixing conditions so that we don't have shear peaks in the system to alter the fate, or to stop altering the fate, or to preventing the altering of the fate of the cells, and of course the efficient nutrient supply. So how does this magic now works? What you can see here is our smallest vessel, the 300 milliliter system on the right side. And even though it's a kind of stop motion video here, you might see that those particles mimicking in this animation or in this video, spheroids or microcarriers work as well. You can also work with single cells. We even did outside the iPSC market with C. elegans, so with complete organisms, but also with cell-free protein expansion systems. Yeah, so the system is completely agnostic to the bioprocess, which works in and works with all bioprocesses. But those particles are soaked in at the top part and then pushed out over the blades to the vessel inner side, repelled there, mixed with others, and then can be soaked in again. So this is, as you see in the middle frame, how the flow pattern is built out when it works in clockwise direction. But the real magic is a gas transfer. And what you can imagine is that we're pushing the process gas over the shaft, here shown at the top part of the video, to the head part of the stirrer. And there the gas is split to each single of those blades. And those blades contain out of dozens of hollow fibers. You can imagine those as plastic macaronis, if you want so. And in the middle, in the lumen part, so the empty part in the middle of the fiber, the gas can flush through. And the molecules of the gas, mostly oxygen, diffuse into the plastic, and from the plastic into the media. We really have no pores here, so it's really a diffusive process, but you can offer a high surface, mostly like 20-fold the head space. And with that technology, we were able to directly presenting the cells the solved oxygen molecules. The cells can consume those molecules, can proliferate, can producing products, and of course, carbon dioxide. And the carbon dioxide, and this is the real beauty of the system, diffuses backwards alongside the concentration gradient. And as we have a rinsing gas in the middle of the fibers, always flushing through, we can really exit the carbon dioxide here as well, stoichiometrically, of what we do inserting with the oxygen. So we don't need to buffer or to add base here for counteracting the acidification of the media by the carbon dioxide. This works automatically with that system here. Of course, you will have lactate production, and you have to add some base, but this is significantly less than in a classical system, which also has a positive effect of the osmolarity. Maybe one word to add here, we can also exchange the bottom part of the vessel with a cell retention system. We call this then the in-situ perfusion. And with this system, we are able to doing, in-situ perfusion. So we can filter out the media, giving in new one, and having this all combined in one bioreactor. When we have a look on the technical data, here on the left side, you see the gas transfer rate as a KLA against the tip speed. And the tip speed here can be used as a rule of thumb or a broad estimation of the energy we have to enter here into the system to get to a decent gas transfer rate for the mixing. And what you see easily is that classical systems like classical stir tank reactors, but also vertical wheel systems are maybe only 1/10 or need 10 times the energy to have the gas transfer we can offer here. If you see this with the direct power input number, and here we don't have so many numbers found in literature, but from the vertical view, for example, this was quite obvious to see in their publications. You see the huge difference between the power you have to put in for having the gas transfer, and they cannot even come close to what we can offer right now with the same energy. We did intense work on simulations of our system to have a better understanding of that, to also getting shear force numbers, and to, of course, optimize the system. Yeah, and here the pictures you see are in a very high resolution, in the meaning that the color selection was exaggerated for having a better understanding of the flow patterns, and we are far below everything what could harm the cells. We have no dead zones here, so we had a close engineering look on this topic with our biology, with our people in the lab, to really understand what are the needs for those iPS cell cultures, and now we finally figured that out. Yeah, so we need laminar flow patterns, we need homogeneous conditions with no dead zones, we need radial and vertical mixing for the nutrients everywhere supplied, but also metabolites getting out of the system. And yeah, of course, the shear force is not just low, but it's controllable. And this means this is also an advantage of the system, actually, that you can also trigger the cells. You don't have to use high shear forces, but you can use that. For example, if you're thinking about differentiation steps regarding chondrocytes or bone tissue, this can be triggered with higher shear forces, but you don't have to make use of that. I already claimed quite boldly that we doing scale up, and what you're seeing here is that we are right now in the 300 milliliter vessel when it comes to our single-use system. This is also true for the 2 liters, and Marco later on will show the results here of that. But we also worked in 10 liters already. Here we did some retrofitting of existing glass vessels, and we're having working prototypes for 50 and even 200 liter bioreactors. The technical upper limitation is regarding the intensification of the process between 1,000 and 2,000 liters. Maybe just for interest, the 2-liter system is something we will roll out at the end of this quarter or the beginning of the next one, and 10-liter at the end of the year, or latestly Q1 in 27. So we're having 2 product lines, just for clarification. On the left side, you see a classical bioreactor controller with pumps for perfusion, for pH control or feed and bleed options. You have the possibility to control the 300 milliliter up to 10 liter systems here, all with one controller. We're having a nice online analytics like DO, pH, and temperature. You can add more things like glucoses. The vessel per se is agnostic to all kinds of analytics. You can also add real-time Raman spectroscopy, for example. We already tried this and it worked very well. It's a fully closed system, so it's a standalone product, and it's of course compatible with our perfusion, but also with classical perfusion systems like ATF and TFF, as you will see later. On the right side, you see our InQ system, which is like the vanilla version of that, or the try-out version, or sometimes we also call this a sophisticated shake flask, as it is a nice little platform, half the size of a sheet, and you just put this into your classical incubator. And here we rely on the atmosphere of the incubator and the temperature. It will soak in the atmosphere of the incubator and give that in the membrane system. So you still have the active aeration over the membrane with all the advantages here, but normally it doesn't come with analytics, especially not with online analytics. But it's a nice thing to try out. It comes with 3 PG13.5 ports, so you even can include existing on analytics if available. So said that, now we're coming to the really interesting part, and I will reach over the microphone to Marco for the second part of our session. And looking forward to your questions later on.
Thank you, Patrick. Hi, everyone. I will gladly now take over this presentation, and we'll shift a little bit the focus into the biological tests that we did. Whenever someone thinks about upscaling and high cell density for clinical application, the first idea comes to mind is the perfusion is the way to go. And most of the cases, it really is. However, perfusion actually can hide some of the limitation of the system that are using. And for us, as we are looking for the best product possible, we want to really go deep into our product and try to find out what are the limitations, how to solve it. And with this in mind, we here decide or plan to perform 4 different experiments looking into the basis of the process itself and try to upscale into higher cell densities and then upscale into the 2-liter vessel. So the first experiment that we are showing here is actually just using, as Patrick said, our ComfyCell InQ, where we place in the incubator, and we used initially just a basic IPS media, try to understand exactly how far can we go in the cell density. From that point onwards, we switched to a more richer media, try to see exactly the limitations on the process itself. And then after that, we really moved to the perfusion, which of course will allow us to really achieve high cell densities, initially in the 300 milliliter scale. And then later on, we already performed the proof of concept in our 2-liter vessel size. Moving ahead, just starting now with our Fedbatch feeding regimen. In our first experiment, where we use traditional essential aid-like media, we started with just 120,000 cells per milliliter and incubated cells for roughly 5 to 6 days and try to see exactly how the aggregation happened and the increase on the cell density or the aggregate size and the cell numbers. And if you can look at the bottom graph where I look into the viable cell density, it was quite interesting to us where we see, as expected, a little bit of reduction on, or a little bit of cell loss at 24-hour mark. However, this recovered quite nicely, and we can see a nice expansion until the 96-hour mark. And then we see a reduction on the expansion rate, and actually a 20-hour mark from the 20-hour mark to the 144, we see actually a slight reduction. As Patrick already mentioned, we have a really high KLA value. So our expectations or assumption is actually the aeration is not the issue here, but the media could be. That's why we decided to test a richer media. But when you look into the aggregate size, which is one of the critical parts for iPSC expansion as using spheroids, we were able to achieve around 200 micron at 96 hours, and this size remained until the end of the run. This actually show that we can produce quite nice aggregates, specific size for further differentiation, and more important than that, the aggregate size distribution remained quite stable with having more or less homogeneous aggregates throughout the entire system, as you can see at the bottom right, when, for example, as an example, the 96-hour mark, with most of the aggregates remain between 176 to 201 micron. Having these, then of course wanted to look into a more rich media, and then we organized 2 different runs or 2 runs with 2 different media. Unfortunately, I cannot share here which media were used or which nutrients and constituents of the media were used, but we basically look into looking different cell densities at the start. We have one of the runs, it was for 5 days only. The run was a 7-day. And actually impressive was that one of the runs, in this case run 2 with media A, at the end of the run, we were able to achieve 3.2 million cells, which I'm going to show now more in detail. With this richer media, we decided then to run for 7 days. We actually, as the first one, got really nice aggregation from the beginning. We can still see a little bit of difference in the aggregate size, but what is really important for us was actually to see that the viability remained quite high throughout the entire run. And more importantly, it was the increase on the expansion, especially from day 5, excuse me, from day 5 to day 7. where actually in this case, we do not achieve any plateau on the cell number, showing actually just the media has a big impact, and the actual bottleneck for this system with active aeration, but limited to the atmosphere of the incubator, we were able to did not see any plateau on the expansion. But of course, not only the cell numbers are the critical part, so we also had a look into the morphology of the cells on the aggregate size, for example. And in this case, we were able to achieve a similar aggregate size with a quite nice distribution throughout the entire time. We can still see, especially in the beginning, some smaller aggregates, but at later time points, we do not see these really small aggregates, showing a quite homogeneous aggregate size distribution. And when you look for the analytics, on this case, the pluripotency, which is one of the most, if not the most critical part on the iPSC expansion, or at the end of the iPSC expansion, we stain for 3 typical pluripotency markers, so OCT3-4, SSEA4, and TRA160. And in all of these 3 markers, we were able to achieve more than 98% expression. Of course, we also wanted to look into some differentiation mark. In this case, just as an exclusion, we use CD15. And as we can see at the bottom right side, we were not able, or we did not have any expression on this marker. Just for this Fedbatch point, so we have some key points to show. So when we use E8-like medium, we were able to observe a plateau at around 200 million cells, despite regular media change. So as I said, so we have regular media change every 24 hours. And this shows actually clear limitation on the nutrient availability for the cells. Whenever you change to more rich media, we're able to really achieve higher cell densities for a Fedbatch, of course, would be on 3 million cells per milliliter. And these numbers were actually do not achieve a plateau there. So showing that actually even with the same regimen and the same media, we might be able to achieve even higher cell numbers. So this just shows actually that oxygen transfer is definitely not a limitation factor at these cell densities, of course. However, of course, if you want to go to higher cell numbers, perfusion needs to be the solution, but it just shows that actually the bottleneck at this scale and at these cell densities is the media and not really the oxygen. However, of course, when you are looking to clinical application, really high cell density is what we are aiming for. So then we decided to shift to a perfusion strategy, try to achieve really high cell densities, the ones required for clinical application. And for this, of course, we wanted to compare ourselves with the highest cell density already published. And in this case, it's the manuscript from Felix Manstein, where it shows that actually could achieve around 35 million cells after 7 days of expansion. And what we really tried to do here was basically just mimic the protocol and see how far can we get with our system. So just really going through, we started with 500,000 cells in 300 milliliter volume, working volume. The media that we used, it was essential media as the first experiment that we showed. And for the supplements, we actually just decided to use Hock inhibitor, while, for example, Felix Manstein work was added different supplements. We work at 70 RPM, which is our traditional RPM for iPSCs for road expansion. And we try to keep the oxygen or the dissolved oxygen at 40%. And the perfusion started at day 1. And this is one of the important parts here. As you see on the right side, the perfusion regimen, which we increase the perfusion rate throughout the entire expansion. And at certain point, we actually started to add glucose and L-glutamine. Jumping into the data, we performed the first proof of concept expansion run, try to mimic or adapt the protocol from Felix. It was not really the same protocol, just as it was the first proof of concept that we did. And we decided that, of course, to start with 500,000 cells as Felix did. And we can see from on the left side, these pictures, we got quite nice aggregate formation. And throughout the entire run, in this case, we did it for initially 5 days. We see that the aggregate size increase, and this can be seen at the bottom right graph, with the increase of the aggregate size to around 500 micron. It is already published, or it is known that the influence of perfusion compared to the Fedbatch has a big impact on aggregate size, which leads to really high aggregate size, which was observed here. This is actually one of the things that we want to work in the next couple of weeks and months, which is trying to control the aggregate size throughout RPM and also initial cell density. However, one thing I want to highlight is when you look into the picture of our vessel, it's barely impossible to see the stirrer, showing that the density of the media is really, really high. But when you look to the cell numbers, and this is what I want to highlight here on the bottom left graph, we really have a nice expansion until the day 5, where we reach around 15 million cells, and also with the same or really high viability. However, as you see in the graph, it's for the last day is in gray, because initially it was only planned for 5 days, so we do not expect that actually this run will go as good as it went. So we do not have enough glucose for this last day. So then we just added media without the glucose, and here we can see a reduction on the expansion rate, however, still reaching around 19 million of iPSCs per milliliter. And this can, of course, be also seen on the fold expansion, where we got around 35 to 37 fold expansion rate, but with a decrease on the doubling time. However, after this first run, we then decided to go with a proper protocol and proper planning. So we had all the media and glucose needed for the 7-day experiment. So as the first one, we decided to start with the same 500,000 cells per milliliter, and you see at day 1 as the same similar data. So a nice aggregate formation. There's still some variability on the aggregate size. However, the aggregates continue to expand and grow in size until the day 7. And again, on the picture from the bioreactor, you can really see, or actually you cannot really see the stirrer, as it's completely covered by really nice aggregates. And in this case, when you look to the viability, remain quite high, above 85%. But more importantly, for us, it was actually the cell density, where we see a really nice expansion, specifically from day 5 to day 7, where we reached around 36 million cells. But because it was running so well, we decided, okay, let's just go for 1 extra day. Let's really push the boundaries and see where our system could see the limitation. However, for this case, as of course, we only plan for the 7 days, and the media exchange is quite high. We do not have enough media for the original 7 VVD as we had from day 6 to day 7, so we have to reduce a little bit. And in this case, we see, of course, that this media exchange was not enough. Nevertheless, it was the way that we got actually the 44 million cells per milliliter just in 8 days, and the viability still remained quite high. When you look to the doubling time and fold expansion, so we see the same pattern. So the expansion or doubling rate remain around 20 hours, except for this last day, where we definitely see a reduction on for the doubling time, and also the reduction on the expansion rate. Of course, the cell numbers is only one part of it. Another part is the analytics, so the aggregate size distribution, as well as the pluripotency markers, as we can see in this graph, or in this slide here. On the left side, you can see again the same pluripotency marker standings that we did in the previous ones. And in this case as well, we have a really nice expression of TRA160, OAK34, and the CA4, and no expression of CD15 was observed. In this case, we really wanted to look into the aggregates, as we saw from the first experiment that we had really high aggregate sizes. So we did actually a cut-through picture here with immunohistochemistry staining for TRA160. And in this case, we can see a nice distribution of fully pluripotent iPSCs throughout the entire aggregate. For this analysis, we want to go a little bit further into the aggregate size and circularity score in this case, and we see the same aggregate size distribution was observed, reaching around 500 micron on day 7. But really important for us was actually to see that the circularity score, which goes from 0 to 3, where 3 is a perfect circularity, 2 is an average, and below 2 it's a poor circularity. And in this case, we have from the beginning until the end of our expansion around 2.8, showing that actually our aggregates, even though they're a little bit bigger than expected or than wanted, they remain quite circular and and they will have quite easy access to all the nutrients required for expansion. After performing then the 300 milliliter vessel size expansion, of course, the next question is how it is when we are upscaling to larger volumes. And in this comes then our next volume size, which is the 2-liter vessel. And of course, when we are working already with this volume size, one of the big issues is achieving or obtaining enough cells to start the run. And for example, for the 2-liter, with the same protocol, we do need 1 billion cells. And the only way to do it, of course, is going through a seed train, which anyway is expected to go if you go for clinical translation. So we started, of course, with our 2D culture using the traditional culture flasks. From these, we detach the cells and transfer into our 300 milliliter vessel size, and we culture for around 3 to 5 days, depending, until we get enough cells for the 2 liter. And after these, then we just harvest the aggregates from the 300 milliliter vessel, get them or digest the aggregates into single cells, and then really start our 2 liter run. And of course, in this case, we prove, I want to show that actually our vessels, they are just simple linear scaling. And you can see on the table on the right side that most of the key process parameters are the same. The cell numbers that we started are the same. Of course, here the media changed because we go from, of course, 300 ml to 2 liter. Media is the same, hockey meter. The only thing that we actually need to change is the impeller speed due to power input. And in this case, we actually have to use half of the RPMs. So from 70 RPM to 45 RPM, but everything is basically the same as the 300 ml. For the perfusion, of course, then it comes one of the limitations in our side as a small company as we are. Cost is a big factor. And of course, if you basically replicate the same protocol, will require a huge amount of media. So we have to adapt a little bit this. And for the 2 liter, we just decided to go for 5 days of expansion, which would show an indication of capabilities of our vessel. And we had to reduce a little bit on the volume change from the last 2 days. Remaining the same on the glucose concentration, also the L-glutamine. So quickly jumping into the data, which are the important part here. From our 2 liter, we were able to achieve a really, really nice aggregate formation at day 1 with basically no cell loss or almost no cell loss from this. And then we can see, of course, an increase on the aggregate size, but also on the cell density. If you look at the pictures and try to look into the stirrer, for example, at day 3, where you can still see the stirrer in the middle of the vessel. But when we go to the day 5, the stirrer is basically invisible. So that's just to show how dense our media is full of aggregates. But most importantly for us, of course, is the cell density. And when we look into the VCD and the viability, so from day 0 to day 5, we're actually able to achieve a really nice expansion, reaching around 11 million cells per milliliter at day 5. And in this case, the viability remained even higher than the 300 ml, except on the day 1, as it is expected, a small drop from the aggregation step, but then remain really high, above 90%. And if you look, of course, to doubling time, it remained really low compared to previous runs that we performed in the beginning, especially for the Fedbatch. But here we have a really nice homogeneity, so remaining around 20 hours for doubling time. And for the fold expansion in 5 days, we're able to achieve around 23-fold expansion. Of course, again, cell numbers are not everything. So we have a look into the morphology of the aggregates and the pluripotency expression. And you can see in the left graph side, in the left-hand side, the same immunohistochemistry staining. And we see from the 2 aggregates that we have there, they are fully pluripotent. So they are expressed at 3160 marker, well distributed throughout the aggregates. And when you look into the expression markers for TRA160, R34, and SEA4, similar data was achieved with above 90% expression, all 3 markers, and no CD15 expression at all. When you look to aggregate site distribution, these were quite surprised, as for the 300 ml, we were able to achieve around 500 at day 5, 6, 500 micron at day 5, 6. But in this case, we're actually able to achieve a smaller aggregate size compared to around 200 micron on day 5, which was quite surprising to us, but ideal aggregate size for further differentiation. And again, in here, when we evaluate the circularity, we're able to achieve a really high circularity score, showing that our aggregates are quite homogeneous throughout the entire time. Of course, and everyone might have in mind the comparison between the 300 ml and the 2 liter, why one was 44 million to the one was 11. So we decided actually just to do a side-by-side comparison. So we actually could really judge the different scales. And what was staggering to us was actually when we look into the cell density as well as fold expansion, of course, particularly from day 4 to day 5, it was a huge improvement in our 2 liter vessel, showing that actually the 2 liter vessel is a more or it was an improvement performance-wise compared to 300 ml. And this can actually be seen as well in doubling time, especially from day 3 to day 5, where have the smaller doubling time compared to 300 milliliter, showing that actually scaling up in this case is not really an issue and could actually improve. Again, I just want to highlight here that these are proof of concept runs. So of course we only did one run in the 2 liter at this moment, but we are looking to it in the near future. Just then to finish the presentation, the next couple of slides, just wanted to actually just bring you and highlight you for what is already shown out there on the cell densities wise. And if you look into the 300 milliliter scale, as we were able to achieve 44 million cells, this is actually 25% higher than the highest numbers that we could find. And this was using a traditional sear tank bioreactor. But when you look into other systems, for example, vertical wheel in this case, or the spinner flask, the difference is staggering, showing that actually our system overperforms anyone else out there. And these just from a small vessel with 300 milliliters, we were able to achieve more than 13 million of iPSCs in just 7 to 8 days, which is remarkable. And maybe even more important is the fully pluripotency expression at the end of it, showing that actually we achieve a really nice cell population at the end. When you look at 2 liter, here the data, it's even more impressive. So when we look just with our vessel, we were able to achieve almost 11 million cells per milliliter, which is more than the double that we have seen out there from the traditional sear tank bioreactor in 2 liters. And of course, if you look into the other ones, as I said before, the vertical wheel or the into 1 liter scale, these numbers even more impressive. And from this, just showing the real number at the end. So just in 5 days in the 2 liter, we're able to achieve more than 22 billion iPSCs. And all these, or most of these cells really show a high pluripotency marker expression. Of course, we are not only looking into iPSC expansion, we are looking, of course, for the differentiation. But as you well know, iPSC expansion as spheroids is not the only setting for expansion. So we also tested different settings. On the left-hand side, you can see encapsulate iPSCs. It was a work done together with Trifrog. And here we're actually just comparing to a traditional tea culture flask. And you're able to, just in 7 days, to achieve 3-fold higher cell densities compared to this tea flask. And even more important that is we got really high pluripotency marker expression at the end of this run. Another setting that we tested as well was, of course, iPSC on carriers, which mimic a little bit more to the expansion, but giving the possibility for upscaling. And in this case, we also were able to outperform not only the T flask, which is well established, but also traditional sear tank bioreactors by almost the double amount of cells, specifically for, or particularly for the sear tank bioreactor. Then, of course, differentiation is then the one of the key parts of it. So we already started to do some work with some partners. You can see on the iPSC-derived organoids, we're actually able to achieve a really nice differentiation just in 7 days from iPSCs to cardiomyocytes, achieving really nice numbers. And also on the lung organoid side of it, we were able to differentiate and expand for more than 35 days iPSC-derived lung organoids. Additional to that, we also are able to already shown really nice neuroprogenitor expansions using carriers or microcarriers here, while the cells remain really high viability. This is not all, of course. We already have quite a lot of work going at the moment. We already have some preliminary data. Unfortunately, we cannot show it here, but at some point in the future, we'll gladly show as well some preliminary data on iPSC-derived microglia differentiation, platelets, and iPSC-derived NK as well. We have already a little bit on the macrophage side, but more will come soon. And we are going now to shift a little bit into the beta cell field as well. And just to finish this presentation, just to some conclusions from my side, we were able to show actually that using a Fedbatch, the key limitation or the bottleneck is actually the media, not the oxygenation, using our system. And we also show that we were able to achieve really, or in our case, the world record on the both 300 milliliter and also 2 liter scale. The iPSC derived or iPSC aggregates were actually quite homogeneous and have a really nice high circularity score. And most important also is actually the pluripotency was fully preserved at both scales. and already have some indication of possible differentiation toward both cardiomyocyte as well as lung organoid. Of course, the work is not over here, and we already have a lot of work to plan for the future. One of the plans, of course, is doing our replicated 2-liter scale to show that actually our data or our process is robust. We are also going to start now expand our differentiation portfolio, specifically into the pancreatic beta cell and macrophage as next targets. But we also need to optimize our culture condition, specifically on the aggregation and the aggregate size. So we are going to work a little bit on that. And of course, after the 2-liter, we are not going to stop here, and we are already having an eye for the 10-liter scale. We are currently, or our engineers are currently developing, and is coming soon as well. With this, I would just like to finish the presentation, and thank you so much for your time, and we are more than happy to answer any questions that you might have.
Thank you very much, Patrick and Marco. This is a very interesting presentation. We have a couple of questions here, and if you have any other questions, feel free to use the Q&A button on the bottom of the screen to ask. First question is about the differentiation. Have you looked at the efficiency of differentiation between the 2D and 3D in terms of the percentage of iPSCs that are converted to differentiated cells and the like?
Yes, actually, that's a really interesting question. With a partner of ours, we're actually able to work this data. Unfortunately, that we cannot really show. We did a macrophage differentiation, and we actually compared to both 2D and other 3D systems that actually the differentiation efficiency was really, really high, actually a little bit higher than the 2D, but specifically for the cell numbers that were achieved, the fold increase compared to the 2D, but the staggering part is actually compared to other systems. We have this data. We hope that we are going to go for a publication together with this partner. So in the future, we'll have this data for you to see.
Thank you. Have you looked at the genetic integrity of in the cultures, especially given the rapid high level of expansion?
At the moment, no. We are going to test. So we froze some of the aggregates at the end of the run, and we're actually going to check for epigenetic mutations. So we'll have a better clue at genetic stability on this. We don't have yet any data on the necrotic. So I know that is a big question or a concern from everyone when you have larger aggregate size. For example, the ones we showed with 500 micron. So we don't have this data yet, but it's something that we are going to look into it in the near future. Yeah.
Maybe to add on the necrotic core problematic or the issue, when we did the organoid cultivation with lung and cardiomyocytic organoids, we reached organoid sizes up to 2 and 3 millimeters, where you normally directly see necrotic cells inside. In our runs, and there are publications to that, which we published this year and the year before. We didn't have that because of the quite well supply of oxygen and nutrients all the time, and because especially in the lung organoids, we had also structuralization in the inner part, so they are not dense. But to see that even 2 to 3 millimeter organoids can be supplied is quite a good hint that this won't be an issue in way smaller spheroids.
Okay, thank you. Have you compared the disposable versus the glass reactors to see if there's any difference between the plastic and the glass when it comes to cell yield or anything?
That's a good question. Actually, we started here with glass vessels because before the injection molding forms, we relied on classical glass vessels, and the results are actually the same. So we don't see any difference here. Okay.
Another question about is it possible to get a configuration with none of the extra sampling ports or anything? I mean, can you do custom configurations of the bioreactors essentially?
Yeah, I mean, we are absolutely open to all kind of configurations here. If there's a need, please reach out, and we will see what we can do here. But normally this is more than possible.
Okay. Have you tried any of the media that are out in the market that are optimized for 3D suspension culture?
Actually, no, that's a really great question. So we haven't tried yet any 3D adapted media in the market. We will test soon some more indicated media, but no, the one that we remain here, because as I said during the presentation, we want to look into the worst case scenario, and not the worst case scenario, but the basic is using the essential 8 media. And then we went from that point onward. So hopefully in the near future, we'll really have more media to test and see the difference in performance.
Okay, have you tried using spheroids as your seed material instead of single cell spheroids developed by a different method as your seed material?
Yes, we actually did, specifically for differentiation. So we did perform or get the aggregates in a Erlenmeyer flask, for example, and then we transferred them into our system, and we were able to continuously grow these aggregates and differentiate them towards the specific cell type. Yeah.
Okay, another question. Do you think there's a theoretical maximum to the cell density? What's the highest cell density you've ever seen with any type of cells?
Oh, okay. So here we can do kind of flex, because just recently we worked with Rentschler on CHO cell cultivation, and here we reached 258 million cells per milliliter in our 2-liter system with an external perfusion system, to be fair. So this is more than possible to reaching extremely high cell numbers.
Was the cell density in that case limited by gas transfer or limited by something else?
No, not at all. So we still had some, let's say, some room here. It's just the perfusion systems. I think in this case it was a ATF starts to block. So it's not possible to, because also you can imagine the viscosity becomes like oil, yeah, like toothpaste. Viscous, it's so viscous like toothpaste. This is nearly impossible to handle then still for the perfusion systems.
Thank you. Somebody asked a interesting question. What is the media change regimen for the Fedbatch mechanism of culture?
Yeah, so for the Fedbatch feeding, we actually performed around 80 to 90% media change every 24 hours. And this can be done actually in different stages or different ways. From one of the experiments, we actually just inserted a deep tube into the vessel, as we have 3 PJ13.5 ports, and we could actually just be able to do the media change there. Alternatively, you can also just skip these, and we actually can aspirate or get most of the aggregates outside and centrifuge it, especially specifically for the first 24 hours. After that, as the aggregates increase in size, we could just stop the stirrer, wait for the aggregates to settle down, and then perform the media change.
Okay, another one is, have you looked at the cost per cell analysis of this method versus other methods when you consider the disposables and the media?
Yeah, I mean, this is quite a good question and hard to answer generally. But of course, as you can imagine, if you have the, it's absolutely process depending. But in general, we hope that we can reduce the cost of cell therapies by 90%.
Okay. Another question about dissociating the cells at the end of the run. If you wanted to dissociate the aggregates into smaller aggregates, have you looked at that?
Sorry, can you repeat?
How you could dissociate the cells at the end of the run to get smaller aggregates, or to separate differentiated cells from non-differentiated cells, things like that?
Yes, that's a really great question. So we actually did not perform a dissociation inside the vessel, as the way we did it was with a seed train. So of course we needed to move into a bigger vessel, so it was not the need for that. We do have the option to, of course, remove through our perfusion system, remove all the media, make the washes, and then digest inside the vessel. We haven't done it. We know it's quite challenging. At some point, then, of course, when you have the single cells, it will become the issue of centrifugation. So we know this is a problem there, but of course, our vessel is ready to interact with other systems that actually can perform the centrifugations for us. So it's something that we can actually look into it in the future. But at the moment, we harvest everything and do the digestion of the aggregates outside the vessel.
Okay. I think that's it on the questions. I would remind everybody that this webinar has been recorded and will be available on our YouTube channel in a few days.
Bob, I'm sorry. I think there was another question popping up with the CHO cell density.
Oh, another so okay, yeah, good point.
Let me just read that. Interested to hear about your CHO density. Again, repeat what the VCD was again. At that high cell density, did you require CO2 stripping? That's a great question. So actually, the cell density was 258 million cells per milliliter in a 2-liter system, and we did not use extra CO2 stripping by micro bubbles or something like that. It worked all over the membrane stirrer. This will be, of course, in publication in the next 2 months. We hope that we can get that published. But if you're having more interest here, please reach out and we can share some more information about that.
Okay, great. If there's no more questions,
maybe we just did our homework right now and had a look into the numbers when it comes to the cost per cell. And if you compare when it comes to iPSCs, for example, with a classical STR, let's say 150 milliliters, we are around 3,000 euros for 1 billion iPSCs. When it comes to our system, we are around 865 euros. So more than 300% cost increase.